Hello, dear learners. Today, I am going to discuss about the immunoglobulin genes and generation of diversity from genes to defense proteins. We all know, and especially the medical students will be aware, that immunoglobulins are a specialized group of proteins which are synthesized by B cells, and it are basically of five types, that is IgM, IgG, IgE, IgD, and IgA. And they have different functions in the body against different type of immune responses. Means that some of, for example, IgM antibody is mostly present in the case of initial infections, or we can say active infection, or when the body encounter any antigen for the first time, where IgG antibody predominates after in the recurrent infections, whereas IgE is responsible for different type of hypersensitivity reactions, and IgA is present in either in the serum or as a secretory part, as a secretory product in the body. Now, we know that all these are made up of globulin proteins, and their functions are also very diverse. And we also know, and each antibody have its own specificity, that it will act against a particular antigen. Means that if infection A is there, then only antibody which is produced against the A will get active, and not the antibody which is active against the B antigen. Now, this diversity is very developed in a very programmed manner in the host cell, and these the proteins we know that mostly are synthesized through the central dogma, that is from the DNA to mRNA and mRNA to protein. Now, seeing the vast diversity or multiple specificity of an antibody, so if we can say, or we can conclude to the point that it may require multiple genes to produce different proteins. Now, this is the very architect of the of our genes product, that they are basically how they will form different type of segments, and how they will develop into different type of immunoglobulin only from a small set of genes, and that is called as the diversity. Now, in this, we will see that how very less number of genes are capable to secrete numerous different type of antibodies having different specificity. So let's start today's topic. So the ability of an individual to produce extremely large number of immunoglobulin molecules, that is approximate 3 into 10 to the power 11, with a relatively small number of genes, has evolved through special genetic mechanism. This occurs because the immunoglobulin genes undergo somatic recombination, which generates an enormous diversity of antibody specificities. Here, if we go to the structure of an antibody, we know that it is a Y-shaped structure. If I can discuss on the monomeric form, and it is developed into two reach chains of a variable and a constant regions. And again, each type of immunoglobulin chain, that is kappa light chain, lambda light chain, and five heavy chains are there, that is lambda H, mu H, I beg your pardon, that is the gamma H, mu H, alpha H, delta H, and epsilon H. So there are two heavy chain, two light chains, that is the kappa light chains and lambda light chains, and five heavy chains, as I have already mentioned in the slide, that is the gamma, mu, alpha, delta, and epsilon. There is a separate pool of gene segments located on different chromosomes. In humans, the multi-gene families are found on the following chromosomes. So these chromosomes, that is for the, if we see here, that is the lambda chromosome is located on the 22, whereas kappa chromosome, that is the two light chains which are synthesized on the chromosome number 2. And the heavy chain family is on the chromosome 14. So, see, as already I have said to you, that how many immunoglobulin, different type of immunoglobulin are synthesized, but the genes which are involved in this formation is very few of them, and they are capable to synthesize multiple forms of antibodies through the different process of recombination, somatic recombination, different recombination takes place for each type of immunoglobulin. Now, each of the three gene loci contains a set of different B gene segments that are separated from the C gene segment. During B cell differentiation, the DNA is rearranged to bring the identified gene segments adjacent to each other in the genome. Electron microscopy studies, Feinstein and Rowe in 1965 indicated that the monomeric immunoglobulin molecule is Y-shaped. The tail of the Y can be clipped by pepsin rejection and represents the Fc portion, that is the fragment of crystallization, while the two arms represent the Fab fragments, which was given by Porter in 1959. The two antigen combining sites are identical and occur in the terminal portion of the fragment F Fab arms. The Fc and Fab portions are joined by a hinge region, which gives flexibility to the molecule in certain classes. By reductive cleavage of human IgG myeloma globulin, two kinds of polypeptide chains are obtained. Now, the larger is called the heavy or H chain, and the smaller the light or L chain. An immunoglobulin molecule consists of two heavy and two light chains joined together by disulfide bridges. There are five classes of heavy chain, as we have already discussed: alpha, gamma, mu, delta, and epsilon, and two types of light chain, kappa and lambda. In any single molecule, both heavy chains are the same class and both light chains are the same type. Both heavy and light chains are made up of homology regions or domains of about 110 amino acid residues. There are two domains of each light chain, a variable region comprising of first 110 to 120 amino acid residues at the amino terminus and a constant CL region. There are four domains in the gamma, alpha, and delta chains, and one BH and three CH in the mu and epsilon chain. The hinge region is replaced by a complete fifth domain, C4. Within each domain, there is a loop of some 60 amino acid residues formed by intra-chain disulfide bond. The amino acid sequence in the variable region, where is found the antigen binding site, is critical for the specificity of the immunoglobulin. So basically, if we see here that the variable region which is present, two regions are there, variable and constant region. So the variable region, which is the antigen binding site is where the antigen bind to the immunoglobulin is very critical part, and that is this that only variable the specificity of the immunoglobulin is mostly decided by the synthesis of the variable regions. Now here two important thing I would like to mention to all of you, that to produce a very large number of different immunoglobulin molecules, that is 10 to the power 6 to 10 to the power 9, without requiring excessive number of genes, a special genetic mechanism, for example DNA rearrangement and RNA splicing, are are used. Okay, so two important functions are there, that is the DNA rearrangement and RNA splicing, and two important genes that encode recombinases. And these the DNA rearrangements are performed by the enzyme called the recombinases. So here we can say that two important step which is important for such diversity in the immunoglobulin is mostly done by the two important process. One is the DNA rearrangement and second is the RNA splicing. So discussing about the DNA rearrangement. So DNA rearrangement is done by the enzyme known as the recombinases, and the recombinases that encode the DNA performed by the two important genes that encode recombinases. Again here, the two important genes which encode recombinases are RAG1 and RAG2, that is the recombination activating genes. So these two important genes, RAG1 and RAG2, gene encode for the enzyme called recombinases, and recombinases is one of the most important enzyme which helps in the process of the one of the most important of one of the important function of the two, that is the DNA rearrangement. On RNA splicing we will come later. So, and if any deficiency in RAG1 or gene defect in RAG1 or RAG2 takes place, so recombinases enzyme is not synthesized by the body in sufficient amount, and if recombinases will not be synthesized, then DNA rearrangement will not take place, and if DNA rearrangement will not take place, then specificity in antibody molecule will not be done, and as such it will lead to SCID, that is a severe combined immunodeficiency. Now here, each of the four then the basic structure of the monomeric unit described is common to all immunoglobulins, that is the IgG, IgA, IgM, IgT, and Ig. However, IgA can occur as a dimer, particularly in secretions, and IgM occurs predominantly as a pentamer, and polymerization is through the Fc portion of the molecule facilitated by an additional polypeptide, the J chain. Immunoglobulins are synthesized by cells of the B lymphocyte line. They arise from the multipotential stem cells originating in the yolk sac, then migrating to the fetal liver and bone marrow, and in adults are found mainly in bone marrow. The clonal secretion theory of antibody formation, Burnett 1959 and Journey 1971, postulates that the immunologically responsive B cells will only respond to a single antigen. The role of antigen is to react with a pre-committed cell, resulting in its proliferation and the generation of a clone of differentiated antibody forming cells. The response of an individual is thus a sum of the responses to all the different lymphocytes contains. Next important here, each pool contains a set of different gene segments widely separated from the D diversity seen only in H chains, J joining, and C gene segments. In the synthesis of an H chain, for example, a particular V region is translocated to lie close to a D segment. Several J segments and a C region, these genes are transcribed into mRNA, and all but one of the J segments are removed by splicing the RNA. So next step, that is the RNA splicing, which is mostly the process, we can say that it is a transcription process. So here what happens, that as the DNA rearrangement takes place, so mostly three, that is the variable portion, diversity, and joining gene segments are together, they come together to each other. That is the V, D, and J gene segments. And after that, what happens? A particular V region is translocated to lie close to the D segment. Of the several V regions, only one type of V variable regions is allowed to come closer to the D segment, and several J segments and a C region. These genes are transcribed into mRNA, and all but one of the J segments are removed by splicing the RNA. During B cell differentiation, the first translocation brings a VH, that is a variable region gene, near a C mu gene, leading to the formation of IgM as the first antibody produced in a primary response. Note that the J or the joining G does not encode the J chain found in IgM and IgA. Note also that the DNA of the unused V, H, and J genes is discarded, so a particular B cell is committed to making antibody with only one specificity. The V region of each chain is encoded by two gene segments, V plus J. The V region of each H chain is encoded by three gene segments, V plus D plus J. These various segments are knotted into one functional V gene by DNA rearrangement. Each of these assembled V gene is then transcribed into the appropriate C genes and spliced to produce an mRNA that codes for the complete peptide chain. L and H chains are synthesized separately on polysomes and then assembled in the cytoplasm by means of disulfide bond or to form H2L2 units. Finally, an oligosaccharide is added to the constant region of the heavy chain, and the immunoglobulin molecule is released from the cell. The gene organization mechanism outlined permits the assembly of a very large number of different molecules. So antibody diversity depends on first, that is the multiple gene segments. So here you can see that there are different multiple gene segments present here. First is the variable region, then D region, that is the diversity, then J region, and then C region, that is the constant region segments. Okay, so it is formed by the recombination of all these genes, and each of them recombination takes place and it forms a specific antibody. And so there is obviously it is necessary that not only one gene can lead to the formation of an antibody, but it is a process through which multiple gene realign together to form an antibody molecule. Then second is the rearrangements into different sequences. Obviously, when these multiple genes are come together and their different segments, rearrangement is also important, which gives specificity to the immunoglobulin, and the combining of different light and heavy chains in the assembly of immunoglobulin molecules. So here, second most important, third most important is the combining of different, and with this we see that there are L and H chains. The V region of each L chain is encoded by two gene segments, that is a V plus J, and the V region of each H chain is encoded by three gene segments. Remember that V region of L chain is synthesized by V plus J region, that is the variable and J joining region, whereas the V region of each heavy chain is encoded by three gene segments, that is the V plus D plus J, and these various segments are united into functional V gene by DNA rearrangement. So it is also an important, and next important is the mutation. So mutation also plays a very important role in uh, the gene rearrangement, because unless and until mutation will not take place, this translocation activity or shifting activity or realignment will it will not be possible. And fifth mechanism called junctional diversity applies primarily to the antibody heavy chain. Junctional diversity occurs by the addition of new nucleotides at the splice junctions between the B, D, and DJ gene segments. The diversity of the T cell antigen receptor is also dependent on the joining of B, D, and J gene segments and the combining of different alpha and beta polypeptide genes. However, unlike antibody mutations, do not play, unlike antibodies, mutations do not play a significant role in the diversity of the T cell receptor. So either it is antibody development or even the T cell development. So remember that three gene segments are the only gene segments which is responsible for the formation of these cells or cell products, that is the V, D, and J segments. And their rearrangement from recombinations, then splicing through method of RNA splicing, they translate into different type of proteins. Now, next is that here that the structure of immunoglobulin with a highly variable and constant region led to the Dreyer and Bennett hypothesis, in which it was proposed that the genes of the variable and constant regions were encoded separately. Moreover, there are many variable region genes in the DNA of the germline, but only or a few constant region genes. So variable region gene are more because it is the only that part of the antibody which binds to the antigen, and therefore most of the variations or specificity of an antibody depends upon the formation of variable regions. So there are many variable region genes in the DNA in the germline, but only one or a few constant region genes. This split gene hypothesis received much criticism at the time, but later research using DNA hybridization sequencing technique indicated that it was essentially true. The variable region of the light chain, for example, is encoded in the germline by the V and J sequences, and the constant region by C gene. The V segments are separated by a long non-coding stretch of DNA from the J sequences, which are also separated by the intervening sequences from the C gene. The heavy chain gene are arranged in a similar basic pattern, but with an additional unique segment, the D sequence. The number of possible antibodies an individual may produce, the contribution of genetic recombinations, and the possible contribution of point mutation to an individual's total repertoire of possible antibody responses was considered by Dr. M. Weigert in the Institute of Cancer Research, Philadelphia, USA, to investigate possible mechanisms that could contribute to antibody diversity. Dr. Weigert's group have initiated analysis of the structure of the antibodies and the genes that bind to the influenza HA molecule. Now here what happens that the investigation has been concentrated on a set of antibodies produced by hybridomas generated from a single mouse. Antibodies of this set could be shown to be produced for by the same VH and VL genes, yet demonstrated a range of different fine specificities for the influenza HA molecule. By analyzing the V regions of these hybridoma antibodies at both the protein and the DNA level, various mechanisms for generating diversities are suggested. So, Dr. M. Weigert demonstrated through experimental procedures that how this antibody diversity comes in the antibody through the hybridoma technology testing on the hybridoma antibody that is which is produced by the process of hybridization on influenza HA molecule, because influenza have more diversified antigen, and one antibody which is produced against one type of influenza will not give immunity against the second influenza. So they usually, that is a separate topic that is basically based on antigenic shift and drift, which usually occurs on the influenza virus. So by analyzing the V regions of these hybridoma antibodies at both the protein and DNA level, various mechanisms for generating diversity are suggested. One mechanism appears to be recombination between different VH genes, but in addition, comparison of antibodies derived from a single mouse indicate that somatic mutations may contribute substantially to variability, which results mainly from single base changes, but these occur continuously, thereby leading to a sequential accumulation of mutations. And finally, the rate of mutation is high. That is, the present estimate is about 10 to the power 3 per base pair per generation. And the reason for this high rate of mutation is unknown. So preliminary analysis around V region genes after the gene segments have been combined. It has been shown that the high mutation rate is not a feature of the V region gene, nor a consequence of the process of V gene development, but rather than the high rate of mutation is conferred the V region by DNA near constant region genes. The genes of the heavy chains and for each of the light chains are encoded on different chromosomes. Each somatic cell has two sets of chromosomes. One member of each chromosome square connected from the mother and another from the father. The stem cell thus has six chromosomes with genetic informations for immunoglobulin synthesis. In the mouse, there are chromosome pairs 12, 6, and 16. By a process of DNA recombination and RNA splicing, the final differentiated cell in the B lymphocyte series will express functionally one H and one L chain gene of integrated form. And the first event in differentiation involves recombination of the DNA coding of the gene. So, with this, I conclude today's lecture. And to summarize today's lecture, I can say that this is a three important things we have to be remember here. That one is the DNA rearrangement, and second is the RNA splicing, two important method which is responsible for this diversity. Second is the recombination of the recombinase enzyme, recombination which occurs through the recombinase enzyme, which is synthesized by RAG1, that is recombination activating gene RAG1 and RAG2, which helps in the DNA rearrangement. And further, three, four important genes which are responsible, because antibody is mostly made of variable region and constant regions, and B, T, and J gene and C region, C gene region are the important regions which decides which part, which type of diversity or which type of immunoglobulin chain will be synthesized, and even the light and heavy chains, which type of light and heavy chains. Basically, constant region remains the same, and the diversity we see is usually in the variable regions. And so that through this chapter, I would like to say you that you can see that there are numerous type of antibodies which are produced, different type of antibodies against different type of infections. But the gene pool which is involved is very less because millions of antibodies are produced in our body for different infections and different type of uh, pathogen attack. But if different gene will be required, so that number of it will be very difficult for the body to synthesize with different animals. So it is the very architect of our genetic makeup that with few loci present on the gene and on the chromosome, such diversified type of antibodies and even T cells are produced. So today's lecture, I conclude it now, and I hope you have been able to understand this topic. So please do like and subscribe my channel, and I will bring more informative videos, educational videos on this on my topic, and help you in your studies. With this, I say you goodbye to all of you. Thank you.